to move on for our first talk of the session. We will welcome Ishani Mukherjee, coming from the Indian Institute of Science, Education and Research, Kolkata in India. So she will talk today about shoaling in troubled water, as well as the impact of rising temperature on shoal properties, foraging behavior, and metabolism in mixed species fish shoals. So Ishani, the floor is yours. You can start sharing your presentation.
Yes, thank you for the introduction. Yeah.
Yeah, perfect. Just put in full screen and we will be set. Yeah, great.
So, hi, I am Ishani Mukherjee and I'm currently doing my PhD on zebrafish. And today I'll be sharing my recent research where I look at how mixed species shoals respond to rising water temperatures. The title of my talk is Shoals in Troubled Waters, Impact of Rising Water Temperature on Mixed Species Shoaling. So what are mixed species groups? Goodell et al. Has described mixed species groups as moving groups of animals that form through interspecific attraction. So these groups are mostly seen foraging together or they are together to get some benefit in terms of escape from a predator. Uh, this is a picture of a mixed species group comprising two fish species. The ones with the vertical stripe are the Indo-Pacific surgent, which is these fishes, and the ones with horizontal stripes are the Australian meadows. So these species, despite looking very different from one another, are found together in the wild. A recent review shows that studies on fish groups are increasing over the years, but despite this, people rarely address mixed species groups, which is the gray, like if the uh, light gray part is the study on single species shoals, the deep gray part shows the studies on mixed shoals. So we also know that zebrafish, which is a very popular organism in biology, in its natural habitat often form mixed shoals. There are reports of this fish shoaling with other species such as Esomus, which is the one with the single stripe. We can see this particular fish, and the zebrafish has multiple stripes. So we observe such shoals in the wild, and we decided to ask how would these shoals react to increase in water temperature. So what did we expect? We know that fish are cold-blooded, and so rise in water temperature will increase fish metabolism. And increased metabolism can be seen in terms of changes in weight, while some species shrink in response to rise in temperature. Not all species do this. Also, increased metabolism can be observed in terms of changes in muscle glycogen content. So if the metabolism increases, fishes will change to anaerobic modes of respiration and use muscle glycogen. But there are reports which also show that some species use fat to oxidize and get energy. And this might result in increased glycogen content. Also, we expected changes in foraging because if metabolism increases, overall food intake should also increase. And It is also well established that fishes prefer to shoal with individuals having similar metabolism. So we hypothesized that if the metabolism changes, their shoaling tendencies might change, and so they may not remain as a mixed species group when the temperature rises. So we could observe shoaling in the terms of reduction in cohesion or polarization. So now we collected mixed species shoals. So this is the map of India, which shows the distribution of zebrafish shoals, and often along with zebrafish, species like Esomus, Pantius, Raspora are found in the same shoal. So in this particular habitat in Haringhata, we observed shoals comprising flying barb, zebrafish, and Gambusia. And we collected the shoals and brought them to the lab. Now we wanted to condition the shoals into 3 temperature regimes. So how did we decide the temperature? There is literature suggesting that in the year 2100, the areas where zebrafish are found will experience a water temperature of like 31 degrees Celsius for a month at stretch. Based on this, we conditioned mixed shoals to either 31 degrees Celsius, which is the temperature expected 100 years, which is expected in 2100. We also conditioned them to 24 degrees Celsius, which is the temperature which is generally prevalent in this habitat, and the third regime where the temperature changed every 24 hours. It was either 24 or 31 degrees Celsius, and it was random. So how many shoals did we test? We tested 20 shoals of each regime. So this is the setup. We kept around 50 fish in such a setup. We had a temperature control and an aerator. So what did we measure? We measured metabolism across treatments. So we weighed the fish, we quantified muscle glycogen, and we also calculated the number of worms consumed by each individual of a species. So this was done by dropping 18 worms, and we manually checked which species is eating how much food. Now we looked at shoaling across treatment. For this, we kept the shoal in an arena like this. And after this, we video recorded the shoal for 10 minutes. And then we tracked individuals using ID tracker and measured cohesion and polarization. So in terms of in response to rising temperature, did weight differ? We see that weight actually differed. In response to rising temperatures, flying barbs reduced their body weight. But interestingly, Gambusia and zebrafish did not show such trends. Next, we wanted to check whether glycogen differed across the temperature regimes, and we see that in response to rising temperature, muscle glycogen significantly increased in Gambusia. So this was very unexpected because we thought that with rising temperature, glycogen would decrease. But what we found was that some fish species, particularly smaller fish species such as zebrafish and Gambusia, they do not use glycogen as a fuel. Rather, they go for fat oxidation, and there is literature supporting this. So, so that is what we saw, and probably that's why the weight of these fish species, Gambusia as well as zebrafish, did not decrease. Now we wanted to test whether foraging behavior differed across the temperature regimes, and we see that there was no significant effect of temperature on foraging. We think that this is because we gave them just 18 worms. Like, had it been greater amount of food, we could have quantified some difference. And interestingly, Gambusia consumed greater amount of food as compared to the other species. So we think this is so because Gambusia is an invasive species and it's not native to India, but it's found along with the other species. Now we wanted to check whether shoaling properties are differing across temperatures. So this is a heat map. So the center is the position of an individual, and the area around it shows the density of individuals around this particular individual. So the deepest shade of red indicates greatest probability of an individual being in that place. So we see when, in case of flying barbs, when the temperature is rising, the shoal is actually becoming less cohesive. And we don't see any such strong decrease in cohesion for zebrafish. But for mixed shoals, there is a slight decrease. But statistically, none of these are significant. And now we wanted to check how polarization is differing across shoals. So what is polarization? So if two fishes are aligned in the same direction over two consecutive time frames, we call them to be polarized. So while this is a less polarized shoal, this shows a highly polarized shoal. So our question was, did polarization differ? We see that across the temperature regimes, there was an effect of species interacting with temperature, and we see that for mixed shoals, polarization significantly reduced when the temperature increased. So we see as the temperature is increasing, in mixed shoals, they are becoming less polarized. So what does it all mean? We know that fishes are cold-blooded and rise in water temperature changed fish metabolism, but this change was different for different species. And we also see that differential changes in metabolism resulted in some changes in shoaling. So shoaling changed also because of other factors which we didn't test, but metabolism and physiology of the fish is certainly a strong influencer of shoaling. And we think that if we had conditioned the fishes for even longer, then this might have resulted in a more drastic change. And we think that if the temperature was like for 31 for at least a year or so, maybe the mixed shoals would become even less cohesive. So this is an ongoing study, and in the future, I want to check whether how the distribution of individuals are in a mixed shoal, whether the interspecies distances are changing across temperature, and we can also check how the velocities of the fishes are changing. Thank you.
Thank you, Ishani. That was really interesting. While waiting for some question on the chat, I have already some. So like, if you can come back just to your slide before. Yeah, this one. Is there any evidence that an increase in temperature of the water in natural condition is correlated with a decrease of foraging resources for these different species?
Yes. Yes, there is evidence, but mostly for larger fish species. When the water temperature increases, foraging increases. There is
Okay.
a difference in winter and summer foraging tendencies as well.
But like the quantity of resources in the water is decreasing, I guess, when the water temperature is increasing.
Uh, Not really. The quantity of resources is increasing with increase in temperature. So there is more
Okay.
algae and
Okay. Yeah. And also like I was wondering if like you tested uh, how much one species would prefer to actually be in a mixed shoal or not, in the sense
yes,
that maybe like if you put the zebrafish with the two other species, for instance, they are getting more
yes.
stressed. So like they
Uh,
will spend more time on the bottom, having a level of cortisol higher, these kind of things.
no, so under, like in a normal condition in the wild, we observe these species together, shoaling together. And I have actually done experiments where I've seen that zebrafish prefer to shoal with a mixed shoal, as well as like they have no preference. Like when I do a choice test, they show equal preference towards a mixed shoal and a conspecific shoal.
Okay, interesting. So like we received our first question. So it's A.B. George who say, great talk. I'm not sure. So he's not sure if he understood what are your results for the fluctuating treatment means. He say, I guess you did not see differences from 24 degrees. Can you comment on this?
Uh, the fluctuating treatment showed intermediate results between the 24-degree and the 31-degree Celsius. So in that treatment, the temperature was changed every uh, 24 hours. And in most of my results, this treatment has a intermediate value compared to the other treatments. For instance, in this particular graph for mixed shoals, the fluctuating treatment has an intermediate median polarization as compared to the 24 degrees Celsius uh, and the 31 degrees Celsius treatment.
Okay. But I think like we are running out of time for your talk. So thanks a lot, Ishani. That was really
Yeah,
interesting. And I invite everyone, if you have a further question, to just
yeah.
go and talk to her on Discord.